Greetings everyone. In this video, I am going to discuss about structure and function of contractile proteins in muscle fibers. Myosin is a large 500 kilodalton protein found in muscle fibers. The isoform of myosin protein in skeletal and smooth muscle fibers is myosin 2. Each myosin molecule is a hexapeptide protein, which means it has 6 polypeptide chains, out of which 2 are heavy chains and 4 are light chains. A myosin molecule can be divided into head, neck, and tail regions. Each myosin molecule contains two globular heads, which are formed by the amino terminals of two heavy chains respectively. Each head has an actin binding site and ATP binding site. The ATP binding site has ATPase activity. The head region also has 4 light chains, out of which 2 are regulatory and 2 are essential. So, each head consists of 1 regulatory and 1 essential light chain. The neck region has only heavy chains, and the neck connects the head to the tail of the myosin molecule. The tail is long and thin, consisting of two heavy chains coiled around each other. The alpha helical portions of heavy chains twist around each other to form the neck and tail region of the molecule. Myosin, when treated with proteolytic enzymes, trypsin or chymotrypsin, gets cleaved into heavy myromyosin and light myromyosin. The heavy myromyosin consists of globular head, neck, and beginning portion of tail, while the light myromyosin consists of major part of the tail. When heavy myromyosin is treated with papain, it is cleaved into subfragment 1 and subfragment 2. The subfragment 1 consists of myosin heads, while the subfragment 2 consists of neck region and just beginning of the tail region. The S1-S2 hinge is located at the junction of S1 and S2. This hinge may be involved in positioning myosin heads while interacting with actin filaments. The S2 light myromyosin hinge is located between the C-terminal of S2 and the N-terminal of light myromyosin and is 152 amino acids long. During polymerization, 200 to 400 myosin molecules orient themselves with their tails pointing towards the center of the thick filament and their heads towards the ends. As a result, the two halves of the thick filament become mirror images of each other, with a short bare zone of only tails in the middle of the thick filament. The cross bridges on either side of the bare zone point in opposite directions. Each actin molecule is a globular protein of 42 kilodalton and is called the G-actin. G-actin monomers form chains of F-actin, also known as the filamentous actin. The two chains of F-actin wind around each other in a helix. The F-actin helix has a pitch of about 73 nanometer, so that its two strands cross over each other once every 36.5 nanometer. The skeletal muscles have beta form of G-actin. Like myosin molecules in thick filaments, G-actin molecules in thin filaments are arranged such that those on one side of the Z discs have one orientation and those on the other side of the Z disc have opposite orientation. The consequence of the polarized organization of thin and thick filaments is that the cross bridges in contact with the thin filament can act like oars to pull the thin filaments toward the center of the sarcomere during contraction.